How are you, my dear students? Today we are going to start our new lessons, and our new lessons is 2. Here's the 2. The headline is the process of the mitosis cell division. Here's the mitosis cell division, my dear students. I just explained a cell. Here's the cell. I just draw this structure is the basically this structure is I draw just only the plant cell. Here's the plant cell. And also this structure is the cytoplasm, and there's the middle structure is the nucleus. Here's the nucleus, the cytoplasm, the another name is the cytokinesis, and the nucleus, the another name is the karyokinesis. My dear students, in this the topics we just explain about the cytokinesis and the karyokinesis, and the another structure is the mitosis. Yes, the mitosis basically there are the another phase. First phase is the prophase, another is the pro metaphase, anaphase, and telophase. In the beginning structure, I just explained today only the prophase and the pro metaphase. My dear students, these topics, basically this structure is the very important topics for your the JSC exam. Sorry. Lesson 2, the process of mitosis cell division. The process of mitosis takes place in 2 phases. The nucleus divides in first phase and the cytoplasm in the second phase. First phase, the nucleus is the divides, and the another structure is the cytoplasm is divides. The division of nucleus is known as the karyokinesis. My dear students, when I just explain, when I just only the divides the nucleus, the another name is the karyo, C-A-R-Y-O, karyokinesis. And the another structure, when is the cytoplasm divides, then the another structure is the cytokinesis. Now, the division, the cytokinesis and the karyokinesis. Mitosis is a continuous process in which the karyokinesis, division of nucleus occurs first, and then cytokinesis, division of cytoplasm takes place. Before starting karyokinesis and cytokinesis, the nucleus of the dividing cell need to take some preparations. These preparations, another name is the interphase. This intermediary stage between the 2 consecutive cell division is known as the interphase or resting stage. Now, we are going to start. The next phase is the division of nucleus or karyokinesis. The nucleus of the dividing cell passes through a continuous complex process and complex karyokinesis. A cell division is a non-separable continuous process. It is not right to divide them into different stages. Still, for the benefit of description, it is divided into 5 stages. These are, again I also repeat, first structure is the prophase, second is the pro metaphase, third is the metaphase, anaphase, and telophase. Now, our only the selected topics, here the selected topics is the first structure is the prophase. What's the meaning of prophase? Basically, prophase, pro, and also the primary phase and also the divide stage. This is the longest stage of mitosis cell division. At this stage, the following events occurs. First structure. Yes, this structure is the nucleus. When I just draw this structure, please everyone see the books and also the boards. This structure is the pin-shaped structure. It's the another name is the nucleolus. And another structure, this structure is the nuclear membrane, and thin-like structure, another name is the chromatin. It's also the mother cell, first cell, and also the mother cell. The early phase, early prophase, here's the early prophase. First structure is the nuclear membrane, and also the chromatin is going to the chromosome. Now, in the chromatin is going to the chromosome, early phase, and also the late phase. Here's the late phase. This structure is the chromatid. When they are divided, again I also repeat, when this structure basically thin-like structure, another name is the chromatin. when they are basically thick and also the thick-like structure. This structure is the chromosome. When the chromosome is also the first, this structure middle layer is centromere and they are divided into four divides. Another name is the chromatid. So first is the chromatin, second is the chromosome, and third is the chromatid. Now I just explained about these characteristics about the prophase. The nucleus become bigger in size. 2. Due to continuous dehydration and the dehydration, nuclear chromatin condenses to form visible thread-like structure known as chromosomes. When they are become from the dehydration or also the minus H2O and also the reduces the water and also the water means H2O or the dehydration. Again I also repeat. Due to continuous dehydration, nuclear chromatin condenses to form visible thread-like structure known as chromosome. Now, I just draw this structure is again, this structure is the thin and also the condenses to form visible thread-like structure. This structure is the chromosome. Then, and it's also the another topic is the early prophase. Now, then each chromosome divides longitudinally into two chromatid. Here's this structure. Where the centromere is come from and also the come from basically the nucleus. When centromere, again I also explain, when the centromere is the come from the chromosome's middle structure, then they are divided into, this structure is the four structure and another name is the chromatid. Then each chromosome divides longitudinally into two chromatid. The sister chromatids are joined together at their centromere. Sister chromatid. This structure is the first structure is the chromatid and another structure is the chromatid. And my dear students, in the biological language, the another name is the sister chromatid. So, I just explained about first structure is the finger prophase and again I also repeat, first structure is the nucleolus, it's also the visible. Second structure is the nuclear membrane and it's also the visible and the chromatin and also the thin-like structure. Again the early prophase, here's the early prophase. We also draw the nuclear membrane. This round-shaped structure is the nuclear membrane and the thick-like structure, the chromatid, it becomes from the chromosome. What's the late prophase? Here's the late prophase. I just draw this structure is the chromatid and this structure is the generating nucleolus. And it's also the visible and the middle structure is the centromere. And then we are going to the next topic, lesson 3. First structure is the Pro Metaphase. Here's the Pro Metaphase, my dear students. Please everyone see this, the Pro Metaphase. Here's the Pro Metaphase first structure. This structure is the pole. Basically there are two poles. Is the North Pole and also the South Pole. And another structure, please everyone see the words. This structure is the spindle apparatus. Basically they are also the spindle apparatus and they are also the chromosome and the degenerating nucleolus, my dear students. When the nucleolus, the prophase stage, they are also the visible, but in the Pro Metaphase structure, they are the not visible. Basically, they are the degenerating nucleolus. And another structure is the nuclear membrane. Basically, this structure and also the thin-like structure is the nuclear membrane. They are also the degenerating nuclear membrane. And the figure is the Pro Metaphase. And we also select basically two categories select. It's come from the plant cell. My dear students, now we are going to the next topic is Pro Metaphase. Nuclear membrane and nucleolus also disappears. Dear students, nuclear membrane, here's the nuclear membrane and the nucleolus. they at the disappears. At the end of prophase, spindle apparatus is formed by the accumulation of some protein fiber in the cell. Yes, this structure is the spindle apparatus is come from the south pole and also the north pole, and they are also the formation of the protein. The middle area of two poles of the spindle apparatus is known as equator. When the middle area, this category and this structure, the another name is the equator, and the equator, the fibers of the spindle spread from one pole to another. These are known as spindle fiber. Please everyone remarks this the line. The fibers of the spindle spread. The fibers, this structure is the fibers like shape. Fibers, spindle of the, again I have to repeat, the fibers of the spindle spread from one pole to another. This one pole to the another, they are joined, and this structure is the spindle apparatus, and it's come from the protein. Equator, the fibers of the spindle spread from one pole to another. These are known as spindle fiber. In animal cells, in addition to formation of spindle apparatus, centriole divides earlier and reaches the two poles from where star fibers radiates. My dear students, I just select basically two phase. First phase is the prophase and another structure is the Pro Metaphase, and we are also going to explain about the just only the karyokinesis. Here's the karyokinesis. Basically, there are 5 phases. 5, again I also repeat. I just explain the prophase and then structure is the Pro Metaphase. Please everyone draw this structure and the figure and also the labeling. Today I think that's all, my dear students. Thank you.